Hello everyone, my name is Venkat Sai Dheeraj. I am studying biotechnology in KL University, Vijayawada. So today I am here to explain you about the isolation of a DNA from a given sample. Now, let's come into the introduction. Here, the DNA isolation is a process of purification of DNA from sample using a combination of physical and chemical methods. It means by using the combination of physical and chemical methods, we can purify the DNA from a given sample. The first isolation of a DNA was done in 1869 by Friedrich Miescher. He was the first founder of isolation of DNA. Methods used to isolate DNA are dependent on the source, age, and size of the sample. In general, they aim to separate DNA present in the nucleus of the cell from other cellular components. The isolation of DNA is needed for genetic analysis mainly, which is used in scientific purposes, medical purposes, and forensic purposes also, etc., which is like used in labs like that. Scientists use DNA in number of applications, such as introduction of a DNA into cells and animals or plants, or for diagnostic purposes. And in medicine, the latter application is the most common thing. On the other hand, forensic science needs to recover DNA for identification of individuals. Like for example, nowadays it is the most common incidents. Everywhere it is happening, like rapes, petty thieves, accidents, or war victims, etc. And paternity determination and plant or animal identification also it is useful. Next, presence of proteins, lipids, polysaccharides, and some other organic or inorganic compounds in the DNA preparation can interfere with DNA analysis. It means the organic compounds or inorganic compounds and some other compounds like it is useful, means it can interfere with DNA analysis method to prepare the DNA. They can also reduce the quality of DNA, leading to its shorter storage life. Next, now let's come into the source. Here, the sources for DNA isolation are very diverse. It means the common sources for DNA isolation includes blood, hair, sperm, bones, nails, tissues, blood stains, saliva, buccal swab, it means like cheek swabs, epithelial cells, urine, and paper cause, which is used for sample collections, bacteria, animal tissue, or plants tissue. Next, basically, it can be isolated from any living or dead organisms. The storage samples can come from archived tissue samples and frozen blood or frozen tissue and exhumed bones or exhumed tissues and ancient human, animal or plant samples. Next, now let's come into the procedure. Here, the isolation of a DNA basically consists of four major steps. It means the first step is preparation of a cell extract, and the second step is purification of a DNA from cell extract, and the third step is concentration of DNA samples. Next, and the final step, fourth step is measurement of purity of a DNA concentration. Now, let's look into the first step. It means preparation of a cell extract. How to prepare the cell extract? Here, uh, to extract DNA from a tissue or a cell of interest, the cell have to be separated and the cell membranes have to be disrupted. It means to prepare the DNA extract, we have to take a sample of a tissue or a cell. We have to separate it, and then the cell membrane has to be disrupted. It means it has to be removed. The extraction buffer helps in carrying out these type of processes. Having lysed the cells, the final step in the preparation of a cell extract is the removal of insoluble cell debris. It means the removal of insoluble cell debris is the final step in the preparation of cell extract. The cell debris and partially digested organelles, etc., can be pellated by centrifugation, leaving the cell extract as a reasonable clear supernatant. It means after the centrifugation, the cell debris and partially digested organelles, etc., these are formed as a pellet, uh, which are formed under the tube. Next, the cell extract is formed into the supernatant, which is a liquid type, clear liquid. Next, chemicals such as EDTA, it means ethylene diamine tetraacetate, which removes the Mg2 plus ions that are essential for preserving the overall structure of a cell membrane, and SGS, sodium doxyl sulfate, which aids in disrupting the cell membrane by removing the lipids of a cell membrane or included in extraction buffer. It means the EDTA solution, which removes the Mg2 plus ions, is essentially helpful for preserving the overall structure of a cell membrane. And the SGS solution, which helps in disrupting the cell membrane by removing the lipids. It means by taking up the lipids in the cell membrane, it can remove the cell membrane, and it includes the extraction buffer. Next, let's come into the second step, purification of DNA from cell extract. In addition to DNA, the cell extract will contain significant quantities of a detergent, proteins, salts, and reagents used during cell lysis step, and RNA. A variety procedure can be used to remove these contaminants, leaving the DNA in pure form. Means the various types of procedures are used to remove these contaminants, and it leaves the DNA in a pure form. Next, the most commonly used procedures are ethanol precipitation, phenol chloroform extraction, and mini column purification. Here, now let's come into the first step, ethanol precipitation. Here, the ethanol precipitation usually by ice called ethanol or isopropanol. Since DNA is insoluble in this alcohol, it will aggregate together, giving a pellet upon centrifugation. It means the DNA is insoluble in this type of alcohols, and it gives a pellet type of form after the centrifugation. Preparation of DNA is improved by increasing of ionic strength, usually by adding sodium acetate. It means by adding the sodium acetate, the precipitation of DNA has increased its ionic strength after adding the sodium acetate. Next, the second step, phenol chloroform extraction. The phenol chloroform extraction, in which phenol denatures proteins in the sample. It means the extraction of phenol chloroform in the denatured phenol in the sample proteins. After centrifugation of the sample, denatured proteins stay in the organic phase, while aqueous phase containing nucleic acid, which is mixed with the chloroform, that removes the phenol residues from the solution. Next, the third step, mini column purification, means the mini column purification that relies on the fact that the nucleic acids may bind to the solid phase, which is dependent on the pH and the salt concentration of the buffer. It means by mini column purification, the nucleic acids may bind to the solid phase like silica and others like etc. By depending on the pH and the salt concentration of the buffer. Next, coming to the third step, concentration of DNA samples. The most frequently used method of concentration is ethanol precipitation. And in the presence of a salt and at a temperature of a minus 20 degrees or below, the absolute ethanol will efficiently precipitate polymeric nucleic acid. Yes. With a concentrated solution of a DNA, one can use a glass rod to pull out the adhering DNA strands, while for dilute solution, precipitated DNA can be collected by centrifugation and it can be redissolved in an appropriate volume of water. You can see here in this picture, we have a 4 steps with the 4 test tubes. In the first step, the cells are lysed using a detergent that disrupts the plasma membrane. It means the plasma membrane was disrupted, it means it is removed. In the second step, in the second test tube, the cell contents are treated with protease and RNase. The protease which is used to destroy the protein and the RNase which is used to destroy the RNA. And in the third step, in the third test tube, you can see the cell debris is pelleted in a centrifuge. The supernatant, which is a liquid form containing the DNA, is transferred into the clean tube. It means the cell debris, which is insoluble, is formed as a pellet after the centrifugation at the bottom of the test tube. And the liquid containing supernatant, it contains the DNA, it is transferred into the clean test tube. You can see it in the fourth test tube. In the fourth stage, the DNA is precipitated with ethanol. It forms viscous strands that can be spooled on the glass rod. It means the DNA which is precipitated, it can be easily taken by the glass rod. The DNA can be easily taken by the glass rod. Next, coming to the final step, fourth one, measurement of purity of a DNA concentration. It means the DNA concentration can be accurately measured by the UV absorbance spectrometer. Means by using the UV absorbance spectrometer, we can accurately measure the DNA concentration. The amount of UV radiation absorbed by a solution of a DNA is directly proportional to the amount of cell DNA sample. Usually here the absorbance is measured at 260 nanometers, and at this wavelength an absorbance of 1.0 corresponds to 50 μg of double stranded DNA per 1 ml. Next, with a pure sample of DNA, the ratio of absorbance at 260 nanometers and 280 nanometers is 1:8 ratio. Uh, the ratio of less than 1:8, it indicates the preparation of contaminated. It indicates the preparation is contaminated, like by either with protein or with a phenol. Um, Next, here in this picture, you can see the samples we have taken: blood, yeast, plants, animals, and bacteria. By taking the samples from them, this is uh, lysated. It is lysated, and the DNA binds into the resin in the left side, and in the right side, the DNA is binding with filter. And finally, we get the purified DNA. Finally, the purified DNA is extracted. And finally, thank you. So, this is about the DNA isolation. If you like this video, please like, share, comment, and thank you.